This particular figure is showing a table called the genetic code. And you'll remember that messenger RNA contains the nucleotides that will subsequently specify which amino acids go into the protein. And one of the things scientists struggled with is when they learned about transcription and they knew about messenger RNA, the question was, how is the information to specify amino acids stored in the messenger RNA? And there were a number of classic experiments that were done to reveal the fact that a particular amino acid is specified by 3 consecutive nucleotides in the RNA. So the idea is if 3 nucleotides specify a particular amino acid, then how many particular combinations of 3 would be available? And so if you think about this conceptually, the probability of getting any particular RNA nucleotide in the first position of the 3 would be 1/4 because there are 4 types of nucleotides. The probability would be the same for the second as well as the third position. So the probability of getting any particular combination of 3 nucleotides would be 1/4 times 1/4 times 1/4, which is equal to 1/64. So there's 64 different possible combinations of 3 consecutive nucleotides. Those combinations of 3 consecutive nucleotides are referred to as codons, and each codon represents an amino acid. And that information about which codon represents which amino acids is contained in this table. So let's take a look at the structure of the table and understand how it's used. So in the column first letter, we have our 4 RNA nucleotides, U, C, A, and G. Across the top, we have our second letter of the codon, and again, U, C, A, G. And over on the other side of the table, we have our third letter of the codon, again, U, C, A, G. So, for example, if you have a particular codon, let's say UAC, the question would be which particular amino acid does that correspond to? So the first letter was U, that puts me in this row. The second letter was A, that puts me in this column. So right now I'm down to these 4. My third nucleotide was C, so that puts me in this portion, which means the particular amino acid coded for by UAC is tyrosine. We could do the same thing with the codon CCC. So my first letter is C, I'm in this row. My second letter is C, which puts me in this column. And my third letter is C, which means the particular amino acid coded for by that codon is proline. Now, you'll notice that there's what's called degeneracy to the table, which means that more than one codon can code for the same amino acid. And you might think, why would this be beneficial? And the answer is considered to be the flexibility. In other words, you could have a mutation in the third position of the codon, and yet you would still get the same particular amino acid. So we said that there are going to be a total of 64 possible codons. So here are my 64 possibilities. Now, you'll notice that 61 of those code for a particular amino acid. However, there are 3 codons, UAA, UAG, and UGA, that correspond to stop codons. These are the codons for which there is no amino acid, and they signal to the ribosome, which is the structure coordinating translation, they signal to the ribosome to terminate translation or to stop translation. The other codon that you'll notice is AUG, which represents methionine, and this is also called the start codon because the ribosome will initiate the process of translation when there's an AUG present immediately after the portion of the mRNA that tells the ribosome, hey, the section that specifies amino acids is coming up right after me. So this is how you use the genetic code. Now, I will never ask you to memorize which codon goes with which amino acid and so on. I'll always provide this table for you on a test to use. So it's not that you have to memorize it, but you have to understand how the particular table is helpful.